All righty. Well, welcome everybody and welcome to our Build a Cell seminar. Thank you, Johannes, for presenting today. And I will let you introduce yourself and present all of your research and everything you've been working on. Thank you so much, everybody.
Thank you, Carly, for the kind words and introduction. So I was approached to give a talk here, and then I could have given the usual things that work. But what you will enjoy today is actually things that don't work. But I thought it was kind of fitting because this project is one of my favorites. It has been with us for over 6 years now, and you will not see many things that work. But then again, I think that is much more realistic than me saying, oh, look at this great publication, it worked. So I hope you will enjoy this. And the talk is called Eternal Cell Life ? Without replication. Okay. The introduction, and I start with the I in introduction. So hi, my name is Johannes. I did my PhD in microbial biotechnology in Greifswald, Germany, which is a really, really small town up here on the Baltic seashore. But it happens to be actually one of the oldest universities of this world. So it has been founded in 1456. And you can see it has very nice surroundings. I also then started my first independent group there. It was a so-called junior research group, and we were trying to produce, I did actually manage to produce so-called drop-in biofuels. Then I moved on to a junior professorship, which is kind of an assistant professorship without tenure. And you can already see to Darmstadt, and you can already see that I don't have nice pictures from Darmstadt. It's okay there, but it's not really as picturesque. And there my group dealt with computer-aided synthetic biology. And since 1.5 years now, I'm a tenured associate professor at NTNU in Trondheim in Norway. And again, the pictures indicated it's really, really, really nice here. So I urge everyone to come. It's really nice to live here and a very good life. So what is our research? Our research is very applied. So we develop and apply microbial cell factories. And for this, we have a variety of platform organisms, and one of them is a yeast, and the other one is a bacteria. And the first one I shortly want to introduce is here our Yarovia platform, and we use this for lipid and hydrocarbon production. So Yarovia lipolytica is a so-called oleogenous yeast. You can see here it can accumulate, so this red stain is null red stained lipids, so that it can accumulate large amounts of lipids, and we do genetic engineering with this, but we also do bioprocess development, and then look what we can do with the products. So this was kind of my starting place in Greifswald, and we still continue working with that. The thing we did then in Darmstadt was to set up a automation platform. So I was tasked, and I did set up this automation platform, which could do automated cloning and strain characterization. And I had to leave this behind in in Darmstadt, and I hope you see me smiling now, because while it was definitely nerdy and fun to play around with this, it also was so frustrating because constantly things did not work, and it was super expensive to run, and you needed a lot of staff, which of course was not funded. So it was fun. I learned a lot, but I also am not happy or not unhappy to have lost it. Another thing we did there that is maybe more relevant to some of this audience here is we also developed the so-called HyperExpress platform, which combines DNA assembly and cell-free protein synthesis. And in the end, we could do all of this in a little above 3 microliters and in a one-pot reaction. But the main thing I took from there, and this is the small advertisement section, is that really what robots can do very well, but what humans cannot do so well is remember where to pipette what. And so we developed this free tool. You can use it for free on PipetteShow.de, so you can design your pipetting workflows and then transfer them to a tablet, put your microtiter plate on the tablet, and it basically tells you where to pipette what. Okay, the other platform we are using a lot is our Bacillus platforms, and there we mostly work with Bacillus subtilis. And what we do here is chassis development. So we are very interested in how can we reduce the genome so the organisms are more fit for industrial processes. So it's not really the basic question, what is the minimal setup for life, but what do we need to take out of the cells in order to make them more robust for industrial processes? And I always say the reason for this is that our cells are actually used to constantly being in the environment, constantly changing conditions, and something constantly tries to kill them. And so they get kind of paranoid. And now we put them to the lab where everything is nice and same temperature, nutrients. And so they are not really good at dealing with that. So they constantly want to protect themselves. And once you kick out these functions from the genome, they become actually much more robust in bioprocesses. The thing we mostly do here in Norway at the moment is working on our spore display platform. So Bacillus subtilis can sporulate, and we can kind of program the surface of these spores. So we can add additional proteins or change the surface proteins properties. And there we do a lot of work with at the moment. But I want to talk mainly about this eternal cell, life without replication. So just a short introduction. We do this with Bacillus subtilis. Bacillus subtilis is a gram-positive model organism. It's a major industrial platform organism, and it's a so-called master of cell differentiation. And what does that mean? It can differentiate into all sorts of different subforms. And one that we use is important for what we're doing is that they can become competent, so they easily take up DNA from the outside. So the genome is very, very easily genetically manipulated. And they can form endospores, and I will come back to this in the very end. Okay, so what is the project about? It started with a very nice grant call from the Volkswagen Foundation, which is a foundation in Germany. And they just asked, it was just called Life. And they said, please submit proposals that deal with basic properties of life. And that was all they asked for. So they got lots of applications, and we were lucky to be among the funded ones. And of course, we also began with the question, what is life? And there's a variety of definitions of life, but one of them is from the biologist Wilhelm Roux, and he gave life these 5 characteristics: so metabolism, growth, adaptation, response to stimuli, and reproduction. And if you break this down to what it could be the molecular level of this, you basically see that there's no reproduction without DNA replication. So the question we asked ourselves is, can we create a population of functional cells which are unable to replicate, and are those cells alive? This was the official question. Another question for me as a biotechnologist is, can we have cells that only produce and do not reproduce? Because in biotechnology, when I feed the cell energy and C source, it mostly reproduces with it and doesn't really only produce. So how do we want to achieve this? We basically want to targeted DNA replication. And the purpose, of course, is duplication of DNA stored information. And the regulation actually solely is done via the initiation of replication. So we have this origin of replication region and the terminus region. And when the cell wants to replicate, it needs to melt its double strands, so to make space for the replisome, so that the whole replication machinery can have enough space in there. Then it's replicated, and as soon as we have 2 copies, the cells can start dividing. So there's control mechanisms that really I have a second copy of my chromosome. So what did we say we do? We said we simply, and the simply came rapidly back to us, we simply cut out the origin of replication. So we use genome editing tools to cut out the origin of replication. And how we envision this process to be is we grow the cells to a desired optical density, so we have enough cells. Then we induce the OVC excision through Cas9, so we wanted to use CRISPR-Cas9 systems. And this will lead to a fluorescence. I will show you why in a moment. And then we can use cell sorting and get a wonderful, huge population of eternal non-replicating cells. This is just the process with the FACS. So we would use a cell sorter that would sort our eternal cells into one pot and the wild type cells into the other one. So we built a genetic construct that looks like this. We put a GFP under a constitutive promoter, so it's always on. But this GFP is split into 2 halves, and they share a large homology region. Now, once we cut with the Cas9 system, we of course get a double strand break, and this is lethal to the cells. So they need to repair this double strand break, otherwise they die. And conveniently, we of course offer them a nice homology repair template for them to repair this. So they can use the overlap of this GFP to repair the double strand break, which of course then gives a functional GFP and helps us sort these cells. And this is what it looks like then. So this is some FACS data. I will shortly walk you through this. So first we measure a negative control. So we have cells without GFP fluorescence, set our gate that the machine knows all of this on the left is non-GFP fluorescent. We have a positive control, so the same construct, of course, not in the ORI region because we can't have it there, constitutively expressed into GFP. And then we sort the cells after 24 hours. And it actually worked. This was, of course, over 1.5 years of work to get to this point, but so far this worked nice. So with a 1 day of sorting, we actually get 11,000 eternal cells. There were, just so you know, some of the challenges that we already encountered here. So one of the challenges was, for example, that we need to sort a lot of cells. So usually we look at 20 million cells in order to actually get enough cells, so these 11,000 cells. And the first challenge we encountered is that the company that provided the FACS, so Sony, their software, they never envisioned anyone sorting 20 million cells, so their software simply stopped after 2 million cells. And these things, so they actually had to reprogram the software, which then again took a lot of time. But in the end, we had some cells. And 11,000 cells, we thought, it's okay, we can work with that. Now, the problem with what we are doing here is that we are sorting, or we are selecting against life. So usually whenever we do a genetic manipulation, we give the cells some kind of advantage. So antibiotic resistance, so only the cells that have this change can replicate and grow. What we are doing here, of course, is the exact opposite. So we make sure that the cells that we want to have are unable to replicate. And this gets back to us here heavily because we have what I like to call contamination by association. So we have here our eternal cell, which has lost the ORI and is GFP. But attached to that, we sadly have another cell, which is still able to replicate, meaning this completely takes over the population right after sorting. So they start replicating again, and we cannot really do any science with it. So one replicating cell is one too many. We then figured out methods to get pure eternal cells, but end up with only roughly 250 to 500 pure eternal cells at the moment. We tried all sorts of things to get around them, and I will just present 2 of them. One I already can tell you did not work at all. So we tried to do an antibiotic resistance and fuse it with the GFP, so that at the same time point we do a GFP, we also get some kind of antibiotic resistance. The cells rapidly escape that. Then we went back into literature and found this paper from 1971, which demonstrated that this compound 6-HPU can actively block the active site of the DNA polymerase. So we thought we just try to do that. And we had actually some very nice students from the chemical department synthesize this for us. So it's not commercially available. We then tested it. This is growth of a Bacillus subtilis producing GFP. And this is increasing concentrations of the compound we synthesized. And we can see that it works. It inhibits growth. The problem is it also affects the GFP signal. So what we can see here, so in purple, you see the wild type. All is well as long as we don't add any 6-HPO. This is our control. So we see wonderful GFP signals. But as soon as we start adding the 6-HPO, we get an equivalent signal from the wild type. So it seems to, we don't know, maybe it's autofluorescence or we don't know. So basically, this also doesn't work. What we also then at least did, we could do some time-lapse microscopy, and this was at least a little bit encouraging, and this is also what keeps me kind of going in this project. So on the left, you see the Bacillus subtilis wild type, and on the right, you see our ORIC mutant. And if we progress in time, you see that on the left, the cells are happily growing, and I will go back and forth, while on the right, nothing is happening, which is good. So we already know they obviously don't replicate anymore, but we also know they don't just pop up and die. Another thing we could do, but only very indirectly, is try to detect metabolism. So we used a stain called CTC, which is reduced by an active metabolism, and then it develops fluorescence. So I walk you through these pictures again. So we have our positive control. On the left axis, we have the signal for the CTC. On the x-axis, we have the GFP signal. And this is our positive control. So everything that is above here is alive, and everything that is on the right has GFP. So we then did our negative control, which is just kill off the cells. So this is why we know everything above here is alive. And then we did our experiment again and could, with this, get an indication that it seems that our eternal cells at least seem to have some kind of metabolism. What we, of course, could not do here is any time course, so we don't know what is happening over time. We then did some deep sequencing and found that it seems like the homologous repair of the GFP after the Cas9 cut is not really that robust, so we get some mutations, frame shifts. And of course, whenever this happens, we lose part of the actual yield because we cannot select for cells that do not show GFP. We then tried alternatives to the Cas9 excision. So we tried a flip parser for recombination. So this would flip a promoter in front of the GFP while removing all of the ORI. This did not work at all. And what kind of worked is a system that is internally from Bacillus subtilis. It's called an SPRAB recombinase. So it's a site-specific recombinase that has evolved in Bacillus subtilis. And what we did here is basically have our same construct, but instead of cutting with Cas9, we now have these site-specific recombination sites. We then, of course, needed to develop or find a position in the GFP that tolerates a scar. So whenever you do the site-specific recombination, a scar remains. And of course, this now would be in a coding sequence. So we did some analysis of the GFP structure and found a spot where we can just insert that tolerates the scar, and tested this, and this is also kind of where we are now. So what's the status quo at the moment? We have sporulation coupled SPRAB excision, and I will dwell a little longer on this. So we have these two problems. We have too little yield, and we have this contamination by association. So what we thought of doing now is kind of use the fact that Bacillus subtilis can sporulate. And this is a very short introduction into sporulation. So we have our normal happy vegetative cell, and it's growing. Then it gets some kind of stress and starts dividing, replicating its chromosome. Then it forms a so-called forespore and moves the chromosome into this forespore. And what now happens is that you have a differential gene expression. So you have a different gene expression in the so-called mother cell and in the forespore. And what we are doing now at the moment is we are making sure that the recombinase is expressed only in the forespore, meaning in here, we start removing the origin of replication. And there is no real competition anymore because this chromosome doesn't take part in any replication or any growth or anything anymore. And so in here, we cut out the OVC. Now this is packed into a spore and the spore is released. And what we can do now is actually heat treat all of this because the spores can survive very high heats, meaning we kill off all the other cells that have not gone through this process and have not really removed the ORI because that only happened for the spore. And then the big question, so this is really recent that this seems to work. Now the question is, can they germinate again? So one thing the spore can do once it gets nutrients again is germinate and become a vegetative cell again. And now the question is, it would then, in theory, just unpack its chromosome and be a vegetative cell, which is unable to replicate. Okay, so we are kind of combining now the two problems, or hope to combine the two problems we have, that we have hopefully higher excision rates with a site-specific system, and we can get rid of contaminants by simply heating the solution. First results are okay. So the yield is not really going that high. So this was first experiment we did some last week and actually got to 2.5%, which is great compared to what we had before. But we don't know yet if the germination will work and all of these things. And I think I've probably been much too fast, but nobody minds a talk that is a little too fast. So I would like to thank the team. So especially Yasmin and Silke did a lot. and of course the Volkswagen Foundation, and we had some partners at the MPI in Berlin in this, and the Volkswagen Foundation for funding this. And of course, thank you for your attention. Okay, and now I see there's questions. Okay, anyone who wants to speak to me, or do we just, I can also answer these and just read them out. But first I want to try if there's somebody brave enough to speak.
They might jump in, but I'm just going to give you a reminder to read the questions out loud before you answer them. I
will do that.
Thank you.
Yeah, I don't mind asking mine over
thank you.
over audio. I was curious, if you were to use DCAS and try to target your origin, do you think that that would allow you to halt replication? And, you know, my thought was that it would kind of sidestep scarring or any of these frame shifts that you see or anything like that.
So this has been done, and it works for roughly 6 to 7 hours. And then some cells find an escape route and are gone. So if you don't cut the ORI, it really is just temporary, which could of course be a solution. But what we really want to do is do this permanently. So we call this eternal cell, and maybe I should explain this why. So my weird hypothesis is that, let's put it differently. It's very difficult to look at aging in bacterial cells. There's some cells that actually look different after they have split, but in generally it's like they split in half, and then which is which. There has been, of course, some work that shows that protein detritus can collect on the poles and so forth. But what we kind of want to do once we have these cells is do things like sublethal salt stress, and then let them sit for a long while and see if they can evolve, if they can adapt. So how do they cope with stress when they have lost the whole function of replication and adaptation and survival of the fittest and so forth? So this is why we are not happy with just 6 to 7 hours. And ultimately, of course, I dream of having like a constant producer. So Bacillus subtilis can secrete many enzymes, for example. So my dream is we have a pot of cells which does not replicate. So I do not get into the trouble that I have too many cells for my compartment or for the nutrients I can provide. And they just constantly secrete an enzyme, optimally over years. And so this is why DCAS wouldn't work for us, because it has been shown that it works shortly, but only for a couple of hours.
Yeah, thank you. That makes a lot of sense.
Okay. Anyone else dares to speak up?
Yeah, I want to ask. This is fantastic work. I love it, especially showing all the not working thing is kind of very interesting to me, because we cannot see those things in the literature. So my question is, you use antibiotic resistant gene and GFP fusion.
Yep.
Have you tried just antibiotic resistant gene itself, and then after the combination, you have the intact resistant gene, and that might be easier in some sense?
We tried, and the problem is the beauty of our approach. Our approach has many drawbacks. So, for example, ridiculous yields. But a little bit, the nice thing about our approach is that we never exert selection pressure on the cells. Right? So as soon as if I would use an antibiotic resistance, I put selection pressure on the population, meaning they can find a way out of that, right? So if I let cells grow with low enough antibiotic resistance, antibiotic concentrations, one cell will find a way out of that and will start replicating again. So this is kind of our philosophy behind that. With the GFP, the cell kind of doesn't know that we are selecting it. So it cannot start to evolve against this.
Oh, no. What I wanted to mention is you let cell just grow or survive without selection at the beginning.
Okay.
And then later on, your challenge is basically out of billion different cells, how to select the one correctly recombine.
Yes.
And to do so, you just let population just do whatever they want to do, and then at the end of the day, you just select against antibiotic, chemical a little bit later. So not immediately you adding antibiotics, but you could kind of delay the application of the antibiotics. That might work. That's my kind of feeling.
Yeah, but then how do we get rid of all the other cells that have not done this recombination?
Other cells will not have the antibiotic resistant gene because they will not, you should have the recombination to have the functional antibiotic resistant gene. That's kind of my thinking.
Yeah, I can't tell you why it didn't work so far, but it didn't. We always had escapees.
No. My question is whether that is because of fusion or because of.
Okay. We tested the fusion. We tested the fusion to kind of outside the ORI. It worked.
But without fusion, whether that will work or not? That's my question. No GFP. So forget about GFP.
Yes.
And then you also tested without GFP fusion.
And I think in the long term you will get cells that escape this, because there is a selection pressure.
Oh, so you're talking about antibiotic resistant
developing afterwards by itself. Yes.
Okay, good. That makes sense.
Okay.
Okay.
Okay, then maybe I asked some questions from the chat. This is from Chris Dennett. This might be a stupid question. Of course, there is no stupid questions, but could you arrest replication by inhibiting some cell cycle checkpoint? Would it stop the replication without the need for genetic manipulation? It's kind of what we are doing, right? So the very first checkpoint of replication is this initiation. And you say we do it later. Would it stop the replication without the need? Yeah, but we again get to the point that it would mean exerting. So you might mean without genetic selection, without genetic pressure. Without genetic manipulation, which means again some chemical that would act like as an antibiotic. And then we get exactly the same problem, that we have some kind of selection pressure present, and then the cells find an escape route from that. Okay, I looked at Brandon. Okay, he says the FAX problem is something we run into too. Trying to sort liposomes, we need to sort order of magnitude more liposomes than the software is ready for. Yeah. The nice thing here was Sony actually reacted really cool, and they just rewrote the software for us. So at least we have a Sony FAX. For them, there is no more limit on how much you can sort. You just need to constantly refill the shed tank. But besides that, it can run forever. Then there's a question from Peter Bandel. Could you try similar work with E. coli or other strain, or is there some reason why Bacillus subtilis is special here? So in the proposal, we of course wrote that this the way this ORIC works and the initiation of replication is universal for all eubacteria. So in general, you could do that to every eubacteric microorganism. I'm sure in nature there's some kind of exception, as there's always an exception, but in general you can do that. We use Bacillus subtilis because the genomic manipulation is so super easy. So we can manipulate the genome really easily. So this is why we started with Bacillus subtilis. Now, if really only the spore way works, then we of course couldn't go to E. coli anymore. But the hope is we learn enough with Bacillus subtilis and maybe the spore process, and then can go back to E. coli and try it there again. And I think there was a similar question. Would yeast be a better model for this? Yeah, but okay. Not in the sense that you can cut out the ORI, simply because the eukaryotic genome has ORIs all over the place. So you couldn't, I mean, we are basically failing with one ORI. So this was Dominic asking, sorry. So it wouldn't work with yeast at the moment. Yep. And of course, you said if you target mitochondria, maybe you could arrest replication while keeping bioproduction. I have to admit, I would need to look into mitochondria more. But then, yeah, if we don't have the mitochondria anymore, we kind of have starving, dying yeasts. Yeah, could be an interesting thing. Honestly, I would need to look deeper into it. Okay. And with this, I think I answered all the questions. Okay, then. Do you think an entity without replication is life or not? This is actually one of my favorite points. I don't know. Because for me, so again, I have a biotechnology background, and one thing that is not so, maybe not so important in the US, but very important in Europe is regulation of genetically modified organisms. And one thing I really want to do, once we are able to actually produce something with that, I will go to the authorities and tell them, I am sorry, but this is not a genetically modified organism anymore because it lacks a basic property of life. It is just a sack of molecular machinery. So, and then I'm interested in what the regulatory authorities say about that. We actually did some very nice science communication around all of that, but that would not really have fitted in the frame. So also working together with the philosophy department, trying to determine if this is life or not. So according to the definition of Wilhelm Roux, this is not life anymore. So
you also think that is not life?
No, if you start looking into it, there's so many definitions of life. So I say, according to the definition of Wilhelm Roux, this is not life anymore.
Okay, but I'm getting old, and I probably will lose ability to replicate later, and then I'm not living entity.
But your cells, I hope your cells are still replicating, because I tell you, if they stop, then I tell you we will not have this discussion anymore. So
okay, I just kind of thinking about that question many times, and I wanted to hear from expert, but that is fantastic answer considering GMO thing. I'm with you. I wanted to also develop GMO and then around the regulatory people. So thank you. That's excellent point.
Okay. And I'm happy Kate agrees with me. Okay, are there any further questions?
I am sure there are. We ended on a pretty thought-provoking question there at the end, but we'll let everybody ruminate on that. And if anybody wants to post a chat right now or speak a new question, of course, feel free, but we'll be ending very shortly. Thank you so much, Johannes, for presenting your research and presenting your incredible questions for us to ruminate on.
Thank you for having me.
I'm sure we'll be thinking about it for a long time.
Okay, great. Then I've achieved something. Okay, then